My name is Sherry Xie. I'm a bioinformatician in the Wang L lab at the Icahn School of Medicine at Mount Sinai, and I'll be introducing the ARCHS4 resource developed by our lab. Over the past few decades, RNA sequencing has quickly become one of the most popular gene expression quantification assays as an alternative to microarrays. Consequently, there has been a parallel increase in the number of platforms, workflows, and tools being developed and published for processing or analyzing RNA-seq data. However, many of these tools require both technical skill and large amounts of resources to run, with sequence alignment being the most computationally expensive step in most analysis pipelines. This has led to a gap between the pace of RNA-seq data generation compared to the pace of data analysis. Additionally, the variety of available platforms also means different datasets are not always interoperable, making it challenging to reuse that data for more integrative analyses. ARCHS4, which stands for All RNA-seq and ChIP-seq Sample and Signature Search, is a repository for uniformly processed human and mouse RNA-seq data from the Gene Expression Omnibus, or GEO. ARCHS4 was first published in 2018 to address the need for a comprehensive and standardized RNA-seq data resource, and is still actively maintained and updated by members of the Wang L lab to this day. The most recent data release as of March 2024 contains over 1.8 million samples from over 20,000 different experiments. Uniformly processing RNA-seq data is important to enable more integrative post-hoc analyses, but most data sets published on GEO are not uniformly processed. Additionally, most people do not have access to high-performance computers that can efficiently and scalably run the first few computationally intensive steps of analysis pipelines, especially the sequence alignment step. ARCHS4 reduces the computational burden on users and provides uniformly processed data in one large matrix that can then facilitate many interesting downstream analyses. All RNA-seq data in ARCHS4 is processed through the same pipeline in order to maximize the interoperability of the data and facilitate more meaningful downstream visualizations and analyses. The pipeline operates entirely in the cloud, which reduces the computational cost of such a large workload and allows users to directly access processed data rather than running a pipeline themselves. All of the data is served on a web-based interface, which provides interactive visualizations of the integrated gene expression data, metadata querying of individual samples, and gene pages with predicted functional annotations. The processed data can also be filtered and directly downloaded from the portal or programmatically for users to run additional analyses. While the web-based portal currently only provides visualizations and functional predictions for human and mouse data, uniformly processed data from additional model organisms such as C. elegans or Drosophila melanogaster can be downloaded from the ARCHS4 Zoo resource. Downloadable files include the relevant species-specific alignment files for reproducibility. The ARCHS4 resource can also be accessed programmatically using the ARCHS4 Python package, which includes functions for loading the ARCHS4 data, extracting specific samples based on a metadata term or GeoID search, and running the ARCHS4 normalization and alignment pipelines on the extracted sample data. An API is also available for querying gene expression signatures against the samples available on ARCHS4 and documented on the ARCHS4 help page. In summary, ARCHS4 is a resource that contains uniformly processed human and mouse RNA-seq data for visualization and analysis, with datasets for additional model organisms available for download from the ARCHS4 Zoo. Because all of the data is originally from the public GEO resource and then processed in a standardized pipeline, ARCHS4 supports the reuse of existing data for interoperative or post-hoc analyses. ARCHS4 also reduces costs by taking advantage of a cloud-based alignment pipeline and simplifies data access for users through an easy-to-use web interface, a dedicated Python package, and a public API. The ARCHS4 project is supported by grant funding from the National Cancer Institute Informatics Technology for Cancer Research Program.